Right, thanks very much for this invitation. I should start by saying, Laszlo, great talk. I enjoyed that very much. So I'm happy to be here to give you an update on the research ongoing in the context of the QCRG, the QBI coronavirus research group that we started about 7 months ago. And before I get into that, I just want to tell you a little bit about the scientific vision of the Institute, QBI, and of ultimately of my laboratory, I'd argue as well. What we really try to do is to develop and integrate together different technologies in a very intuitive way. And we focus this integrated suite of technologies on a variety of different disease areas. So it's a disease agnostic pipeline that we've been trying to set up here over the last several years. And in this pipeline, we have mass spectrometry. So we're studying proteins, protein-protein interactions, which you just heard from Laszlo, as well as post-translational modifications. Try to combine that with genetics, CRISPR-based genetics, and then combine that with structural analysis of individual proteins using crystallography, or now more and more cryo-EM. And then we try to combine all this information with chemistry or chemical biology approaches. So it's a disease agnostic pipeline that I said before, and we're focusing it on a variety of different disease areas in the context of these different cell mapping initiatives. So the cancer cell map initiative, so we're focused on cancer, the psychiatric cell map initiative. So there's a big focus on different neuropsychiatric disorders, including autism and schizophrenia. And then also we're using these tools to study the host pathogen interface under the umbrella of the host pathogen mapping initiative. So here the idea is that we're using these unbiased tools to systematically understand how different pathogens come in and hijack and rewire the host. And it is, of course, under this umbrella that we're studying SARS-CoV-2. So here's a group of scientists that have come together at QBI in this QCRG. There's over 40 different labs at QBI connected to this effort. This is a picture of a number of us taken a few days before the shelter in place last March. So in total here, over 40 groups. So we have systems biologists, structural biologists, chemical biologists, computer scientists, virologists, and many more. And we've all come together, I think, in a very unprecedented and exciting way to try to come up with solutions for this pandemic that we're dealing with. So this QCRG got so large, we actually had to break it up into different subgroups, and these groups are focused on different technological areas such as drug discovery. This is led by Kayvon Shokat and Brian Shoyket. Also, there's a subgroup on structural biology led by the QBI fellow Clem Verba and Oren Rosenberg. I'll be discussing this a little bit later, efforts from this subgroup. And then we also have subgroups focused on different biological areas, different processes that the virus comes in and hijacks and rewires during the course of infection. For example, Davide Ruggiero leads the translational group, and then John Gross leads the ubiquitination group. So there's essentially hundreds of different scientists represented in these different groups that are all really working together in a very, I would argue, exciting way. And we're also collaborating with groups around the world, which you're going to hear about shortly. So one of the focuses of the QCRG is to try to derive host-directed therapies. There's a lot of work ongoing in the QCRG and around the world trying to find drugs and compounds that would target directly the viral proteins, and that's great, but we also want to understand how the virus comes in and hijacks and rewires the host during the course of infection. And then, kind of as Laszlo was alluding to, can we come up with drugs or compounds to target the human proteins that the virus needs to infect our cells? And there's advantages to this type of strategy, host-directed therapy. You don't have to worry so much about resistance, right? Because we don't mutate as fast as the virus. We and others have shown that different viruses actually hijack similar machinery in our cells. So if we could come up with a strategy for COVID-19, hopefully that would work for COVID-22 and COVID-24 and other viruses as well. So the advantage here is ultimately to derive pan-pathogenic strategies in the future. There's disadvantages, obviously, there's toxicity issues, but initially what we've been focusing on are drugs that are already FDA approved or compounds in clinical trials that have already passed toxicity issues. So hopefully toxicity wouldn't be such an issue. So the virus itself has around 30 genes or proteins compared to 20,000 genes approximately in each one of our cells. So the virus can't live by itself. It needs our cells, our genes, our proteins in order to live and replicate and infect our cells. So the question is, which genes and proteins does the virus need in order to infect our cells? So we carried out an affinity tag purification mass spec-based approach like we've done in many other disease areas over the last 20 years to identify what we think is a set of proteins and genes that are enriched for the virus needing to infect our cells. So we identified initially over 300 genes and proteins. And then we looked at these and then we tried to identify drugs and compounds that would come in and inhibit at least one of these. And this allowed us to narrow down to 66 different druggable targets corresponding to 69 different drugs and compounds, where we would hope that some of these would actually have antiviral effects in cells. So there's a couple of papers that we published recently that I'm going to be telling you about initially. So the first one is this particular study that I just had on the previous slide, the SARS-CoV-2 human protein-protein interaction map and how we're using it to identify targets for drug repurposing. The second paper I'm going to be telling you about is also a study trying to understand how the virus comes in and hijacks our machinery, but specifically looking at phosphorylation and specifically trying to identify kinases that the virus needs to manipulate to optimally infect us. And I think this is important for a couple of reasons. There's about 500 kinases in each one of our cells. I like to think of the kinases as the master switchboard of the cell because they're pretty much regulating every process. So it would make sense for the virus to come in and manipulate this switchboard, and it does. And the second thing that I think is important is that many of these kinases are druggable. There's a lot of drugs and compounds that inhibit kinases, many for other diseases, including cancer. So there's a wide array of potential targets and potential drugs and compounds that could be brought to bear to combat COVID-19. But initially, I want to tell you about this particular paper. So back in January, when this virus looked like it was going to cause some trouble, David Gordon, a postdoc in my lab, and Gwendolyn Zhang, a technician, pulled this sequence out. This was the first sequence that I guess it was on GenBake, and it was from Washington State. And we started to synthesize these genes. I think we're the first group to synthesize all of the genes, approximately 30 of them. I know there's probably a few more now that this virus has been characterized further, but we started with about 30, and we put affinity tags on each one of them, expressed them in human cells, lysed those cells, purified each one of the proteins using 2x strep, that was our affinity tag, and then analyzed the purified material by mass spectrometry, and then used algorithms that we and others have developed to come up with a quantitative, high confidence, hopefully, SARS-CoV-2 human protein-protein interaction map. And at the time we put this map out on bioRxiv at the end of March, I just tweeted simply from the lab Twitter account that we have these constructs, we have these genes, we'll happy to distribute, no strings attached, no MTAs, there were no lawyers around anyway, which I think was a great thing. And we said, please feel free to distribute, and we'd even pay for shipping. And in a matter of weeks, we actually sent out these constructs to almost 400 labs in over 40 countries around the world. And I like to say that these constructs, these genes, went around the world a lot faster than the virus did. And we're very proud that we were able to send out these reagents to many labs around the world to expedite research on SARS-CoV-2 and COVID-19. So hopefully others could try to use these reagents to help us combat this particular virus. So here was one of the iterations of our protein-protein interaction map, the 332 SARS-CoV-2 human protein-protein interactions, including, as I said, 66 druggable host proteins corresponding to 69 different drugs and compounds. In this representation, the red diamonds obviously are the SARS-CoV-2 proteins. The nodes, the circular nodes, are the human proteins, and if it's orange, we think it's a druggable target. So early on, actually, we got some help from a company that was based in LA. It's based out of Hollywood. This was a connection made to us from the founder of Twitter, Biz Stone and Ron Conway in the Bay Area. They introduced us to Zoic Labs, and this is a company that's well known for making movies, including Life of Pi and Titanic. And they took our data and they took their software and they came up with a very interactive, I would argue intuitive database and website where one can come in and peruse very easily this information. We're starting to put other data into it, as are other groups. So if you're interested in this data and similar types of data, this kind of network data, and other data like structural information, which I'll get to in a little bit, I'd very much encourage you to look at this particular website. All right, so one of the goals here was then, as I said, was to look at this map, identify drugs and compounds, which are in these boxes here. This was work led by Brian Shoykat and Kayvon Shokat at QBI and at UCSF. Here's the one drug that obviously everybody knows about, remdesivir, targeting NSP12, and the question is, are any of these drugs and compounds, which are a mixture of approved drugs in clinical trials and preclinical compounds, would they have any antiviral effects against COVID-19? And at the time we had these, we didn't have the virus propagating here at UCSF or even in the Bay Area. Melanie Ott here at the Gladstone Institute, where I'm sitting right now, She's made some great strides over the last couple months. We're starting to work with her now. She does have the virus working, but at the time, at the end of March, we just did not have the virus in a laboratory setting where we could test these drugs. So what we did was leverage our great network of collaborators around the world, including scientists at the Pasteur Institute. We worked closely with Marco Vignuzzi and Veronica in his lab, and along with Olivia Schwartz and Christophe Danfer. And then also we worked closely with a good friend of mine, a good collaborator for many, many years, Adolfo Garcia Sastra and Chris White in his lab. This is in the Department of Microbiology at Mount Sinai Hospital in New York. They quickly got the virus up and going so that they could test our drugs and compounds. And these were initially the antiviral assays that they used to test our drugs and compounds. At the time, as Lazo was alluding to, we were using African green monkey cells, Vero6 cells. At that time, we didn't know what human cells that we could use. Of course, we're using human cells now, which I'll get to later. But at the time, we had Vero6 cells. We let them grow 2 hours before infection. We added the drugs, then infected 2 different MOIs. It was about 4 times higher in Paris. In each city, the assay was let to go, and you added formaldehyde to kill the reaction. In New York, they used an antibody against NP as their readout for infectivity, and then also did TCID50 assays in several cases. Paris didn't look at the protein composition of the virus. They looked at the RNA component. So there, the readout was qRT-PCR to look at infectivity, and they're also doing plaque assays as well. And at the time we published this work in New York and Paris, they tested almost 2/3 or 50 of the drugs and compounds that we had predicted. And this allowed us to narrow in on a handful of drugs and compounds that we were particularly excited about. There's a couple here linked to protein translation. The first one here is zotatofin. This is a drug from the company Effector. There's a couple of co-founders of Effector here in the Bay Area, including Davide Ruggiero and Kayvon Shokat. And this is a drug that's being used right now to treat multiple myeloma. And it targets a translational initiation protein called EIF4A. And this is important because we identified EIF4H, another translational initiation protein in our protein-protein interaction map, physically connected to NSP9, one of the coronavirus proteins. And this is important because EIF4H is a helicase that directly works on EIF4A. And in both cities, we found it had potent antiviral effects. We now have great data in primary cells, and the FDA has just approved zotatofin for a clinical trial. So we're working closely with Effector to get this expedited in to people. There's another compound, a preclinical compound initially, that we showed had an effect. This was a compound synthesized by Jack Taunton here at UCSF. It's ternatin-4, and this binds to and inhibits a translational elongation protein called EIF1A. This was a connection also derived from our map. And this had potent effects in both cities. And this is important because there is an approved drug that targets the same protein, EIF1A, called pladepsin or Aplidin. This is a drug, ironically, also being used to treat multiple myeloma, and it's from the company PharmaMar. I'm going to come back to this in just a few slides, where this is the drug that we're actually most excited about, and I'm going to show you some data on that shortly. All right, so protein translation inhibitors we're excited about, but then also modulators that are known modulators or predicted modulators of 2 receptors that came out of our map, sigma-1 and sigma-2. These are actually quite interesting receptors that have been studied, although many people still classify them as orphan receptors because we don't know what the natural molecule in the cell is that comes in and modulates these receptors. But several putative or known regulators of these receptors had antiviral effects, including antihistamines, antipsychotics, the female hormone progesterone, anti-anxiety medication as well, and some preclinical compounds. So Brian Shoykat right now is taking the charge on this particular area, and we're finding the best in class, a lot of mouse work to see which ones we should take to the next stage. And a couple of months after we initially released our map on bioRxiv, Nature Biotechnology wrote up a piece, and they had counted 13 different drugs that had been derived from the connections in this paper that were presently being tested in clinical trials for COVID-19. That number has actually been augmented a lot over the last several months, and we're collaborating with some of these groups and watching the majority of them just to see if any of these drugs will have any value in terms of combating SARS-CoV-2 and COVID-19. All right, so the second paper that I alluded to is this one. It's a very collaborative as well, as you can see. It involves many of the same characters that we had on the previous paper looking at the protein-protein interaction network. This is the global phosphorylation landscape of SARS-CoV-2 infection. So what we did here, this was work actually done in Paris, we took cells, and these were, again, VROE6 cells, and infected them with SARS-CoV-2. And then in a time-dependent manner, in a post-infection manner, we were globally looking at changes of phosphorylation in the host using mass spectrometry. We identified a number of sites, both on host proteins and on the viral proteins, that were changing over the course of infection. So we could cluster this data. So for example, in cluster 1 here, this light blue, here's a number of host proteins that become hyperphosphorylated as infection progresses. This is this blue line up here. And in contrast, say these 2 green lines, which correspond to clusters 3 and 4, these are proteins that become hypophosphorylated as infection progresses. So there's a ton of information in here that we and others are driving hypotheses from and testing about how the virus comes in and manipulates these signaling pathways. But one of the things we wanted to do, as I said earlier, was to look at this data and then try to predict the kinases that were being misregulated. And in order to do this, we collaborated with Pedro Beltrao. He was actually a postdoc in my lab a few years ago. He's a professor now in Cambridge at EBI, EMBL. And out of the 500 kinases, he identified about, based on this data, about 10% of them, about 50 of them, that were either up or down regulated in the context of infection. So we have a number here, including casein kinase 2 at the top, that are upregulated, that's in red. And then we have a number here that are downregulated, including CDK1 at the bottom, in the context of infection. So about 50 overall piqued our interest based on this data. And what was great is we have, I would argue, the best scientist in the world to pharmacologically inhibit kinases. That's Keyvan Shokat, just down the hall, and he's got the biggest freezer of kinase inhibitors in the world. So we went and talked to him. Well, we talked to him via Zoom, I guess, but we talked to him anyway, and we gave him the kinases, and then he went to his freezer and identified a number of kinase inhibitors that then we wanted to check and see if they have any antiviral effects, and this is exactly what we did. And this allowed us then to identify that a casein kinase 2 inhibitor, which is in a clinical trial right now, does have antiviral effects. And this is interesting because it also has a physical connection, casein kinase 2 does, to the N protein. And I'll remind you that casein kinase 2 was the most upregulated kinase in our analysis. Here, an inhibitor of CDK also has potent antiviral effects. If you look in red here, pilimide, which is a PI5 kinase inhibitor, is also potent. And there were another set of kinases here that were upregulated that were all in the same pathway, this P38 pathway. And this is important because there's a number of kinases in this pathway. And it's also important because there's a number of drugs and compounds that are known to inhibit these MAP kinases as well, including these 2, which are pretty potent, very potent, I would argue. And what we showed here on the right is that we were using RNAi to knock down these MAP kinases, and we saw a decrease of infection. So this is a genetic validation of the pharmacological inhibition that I'm showing you here on the left. And there's a number of companies now that have been using P38 inhibitors for treating or trying to treat cancer, and a number of them now are segueing to use these in clinical trials for COVID-19. I just want to go in a little bit deeper into this connection here between casein kinase 2 and the N protein. So we found it upregulated, we found it physically connected with the N protein, and then we found a casein kinase 2 inhibitor inhibiting viral infection. So we wanted to look at this in more detail in the context of infected cells. Specifically, we're going to see, does the N protein and casein kinase 2 co-localize using microscopy and in infected cells? So to do this, we collaborated with Robert Gross at the University of Freiburg, and he started to infect these cells and look at them under a microscope, and he called me up, he was very excited, and he said, well, look what's happening in infected cells. You're getting these actin-like protrusions, these are called Filipodia, only in infected cells. And this is important because this is a way for the virus to infect other cells in an ACE2 independent manner. So what could happen is these long stringy protrusions can poke holes in other cells, and then the virus can tunnel through them and infect cells much more efficiently. Other viruses have been shown to do this, including influenza and smallpox, but not to this extent. I don't think this has ever been described. And to my knowledge, no one's seen this type of branching before, which obviously would expedite transfer of virus from cell to cell. And then when he actually looked at where the N protein kinase 2 is actually in the Filipodia, so we think the virus is actually using casein kinase 2 here to generate this Filipodia. We also worked with Elizabeth Fisher at Rocky Mountain Labs in Montana to visualize this a different way using electron microscopy. So you can see here's the Filipodia here, and at the tip here are viral particles budding off the Filipodia. She generated a number of very exciting images for us, including this one as well, where the red spaghetti is the Filipodia and the viral particles here are in yellow. And here's our working model right now. So we think a couple of viral proteins are hijacking different pathways, signaling pathways that are involved in cytoskeleton reorganization. The virus is manipulating these pathways, we think, to form this Filipodia so that virus can transfer from cell to cell more efficiently. So we think that there's one pathway being manipulated by N, as I said, hijacking casein kinase 2, and hyperphosphorylating a number of proteins, which is allowing for an optimal Filipodia formation via reorganization of the cytoskeleton. We also think NSP7 is manipulating another pathway involving Rho, which is resulting in hypophosphorylation of a number of proteins, which again is allowing for Filipodia formation. So we're looking in greater detail here at these pathways, and this is our working model right now. And I just want to end this part of the talk by showing you one other picture, again from Elizabeth Fisher. It's a very kind of a creepy picture here where you have these tentacles coming off the cell, or they look like tentacles anyway. And in here, the viral particles are in blue. And I really like this image because it looks very pretty, and maybe creepy too, pretty and creepy, but it's also biologically revealing as well. And those are the best type of microscopy pictures or electron microscopy pictures that one can take. So there's been a number of press agencies following our work, including the San Francisco Chronicle. And back at the end of June, when we had published these two papers initially, they asked us for what we thought were the top 10 drugs or compounds that we were most excited about. And this was the list. And at the time, there were 3 clinical trials that were starting since the end of June. I think now 7 out of the 10 are now in clinical trials. I just want to go into a little bit more detail into this particular drug, this Aplidin, which is this inhibitor of EIF1A. Again, it's approved for multiple myeloma in Australia, and it's from the Spanish company PharmaMar. So this is some unpublished work. We have this paper that was just recently submitted in collaboration with Adolfo Garcia Sastre and Chris White. So in human cells, this is HEC293 cells expressing ACE2, we can see really great potency with this particular drug, this Aplidin. It's actually sub-nanomolar potency. Adolfo and our collaborators in Paris have screened essentially thousands, if not tens of thousands of drugs and compounds at this time. This is by far the most potent that they've seen. And it's actually 30 times more potent than remdesivir in this particular cell type. We've also looked in primary lung cells. And you can see at 1/10 the concentration of remdesivir, you're essentially killing the virus very effectively. And Adolfo set up a mouse model where we could test these drugs as well. We're actually using an adenovirus to introduce human ACE2 into the mice. And then you can infect them with SARS-CoV-2. And then over 3 days, you're adding different drugs at different doses. And then you're extracting the lungs from these mice and then looking at how much, or looking at viral titers in the lungs. So here with Aplidin, at a much lower concentration than you see for remdesivir, you can see about a 2.5 log decrease of infection, about 500-fold decrease, which is a much better decrease than remdesivir, which is about 1.5 logs. So we're very excited about this particular drug. And a couple of weeks ago, PharmaMar announced positive results of its Aplidin trial. It's a phase 1, phase 2 against COVID-19. And I think they just got FDA approval, or FDA, a stamp of approval, I guess, to start a phase 3 trial. And what's interesting, even more interesting in my mind about this particular drug, it actually comes from this particular creature that is off the coast of Spain. And this is the particular structure of it. And work not done by us, but by other group, actually have a crystal structure of it binding here in color to EIF1A. And data I'm not going to show you, we actually CRISPR in one specific mutation in this binding site in EIF1A. You basically make the cells resistant to Aplidin. So this, we think, is pretty much the only target for this drug. And we're hopeful that it'll have some positive effects. One downside, it has to be introduced by IV, similar to remdesivir, but we think it will provide some benefit to people. And I'm hopeful that many of you will hear about this in the very near future. Okay, so for the last part of my talk, I'm going to discuss some of our other work where we've been looking across different coronaviruses. Obviously, we're in the midst of SARS-CoV-2. These numbers are now obviously outdated, but as we all know, since the turn of the century, we've been dealing with a couple of other coronaviruses, such as SARS-CoV-1 and MERS. So the logic here is, why don't we look back? Why don't we study the trajectory, if you will, or the evolutionary trajectory of these viruses, so that we could potentially come up with something that's pan-pathogenic, so that we'd be better prepared for SARS-CoV-3, because it's coming. So can we look back in an effort to look forward? And this was a paper that was published a couple of weeks ago, and it's very collaborative as well. It's 200 authors from 16 different institutions in 6 different countries. That's how we could move so fast, is because we collaborated. And this is a paper, Comparative Host Coronavirus Protein Interaction Networks Reveal Pan-Viral Disease Mechanisms. And here are the different collaborators represented around the world. So you can see it was a truly international effort, this particular study. And I've talked a lot about Mount Sinai and the Pasteur Institute, but for this study and the 2 other studies that I talked about, there's been a number of other academic institutions that have played pivotal roles in this work, and not just academic institutions, but industry connections as well, big pharma as well as smaller companies. And several of these you'll hear about over the next couple of slides. So for me, it's been fantastic to see this level of collaboration, not just in the academic setting, but between the academic world and industry as well. And the goal hopefully would be to keep these type of collaborations going as the dust settles on COVID-19. And this is a point I'll come back to at the very end of my seminar. So here's an overview of this study. So just like we did for SARS-CoV-2, we cloned out each one of the SARS-CoV-1 proteins and the MERS proteins and put affinity tags on them, expressed them again in human cells, purified them using the affinity tag, and then analyzed the purified material by mass spectrometry to derive these protein-protein interaction maps. In parallel, we were also doing transient transfections of each one of these proteins and using microscopy to determine in which organelle each one of these proteins was localizing to. And I should also point out that we've also derived antibodies for about 15 or half of the SARS-CoV-2 proteins so that we could localize those proteins in infected cells as well to come up with a cellular localization map. And the idea is we're going to merge these data together and then combine this with genetic functional analysis, prioritize it for structural analysis using cryo-EM, and then trying to connect this data to real-world clinical information. Right? So this is an overview of this particular paper that was just published a couple of weeks ago. So as I said, just like we generated for SARS-CoV-2, we generated a SARS-CoV-1 map, 25 different SARS-CoV-1 proteins on this map, over 350 human proteins. Here's the MERS map that we derived, over 20 MERS proteins, almost 300 human proteins. And there's a number of different ways in which you can compare these 3 different maps. One simple way, and a very effective way, I would argue, is just to cluster this data. And what you can see is about 1/5 of the interactions in all 3 maps are shared between all 3 viruses. About 1/4 of them are specific to SARS-1 and SARS-2, and about 1/4 of them are specific to MERS. So we were particularly interested in these 3 groups, and we've highlighted this in this differential network mapping analysis that we did in collaboration with Troy Ideker in San Diego. We'd actually derived this strategy to look at networks across healthy and diseased cells, and we took it and adapted it quite easily to compare across different viruses. The math is on the top left, which I won't go through, but just to remind you, these were the 3 clusters that were most interested, interactions with all 3 viruses, just with SARS-1 and SARS-2, and then just with MERS. And the edges here correspond to the 3 different categories. So if an edge is black, it means we've identified it with 3 viruses. If it's blue, it's MERS specific. If it's red, it's SARS-1 and SARS-2 specific. And just to zoom up on a couple of connections here. So NSP6, we find interacting with sigma R1 only with SARS-1 and SARS-2, not with MERS. So this is obviously one protein that we've been focused a lot on that we think is connected maybe just to SARS-1 and SARS-2 and not MERS. In comparison, if you look at the N protein, there's a number of black edges. So these are connections that we see with all 3 viruses, including a connection with casein kinase 2. And remember, I told you about this drug. It's a casein kinase 2 inhibitor, which has antiviral effects against SARS-CoV-2. Based on this map, we think it would have effects against SARS-CoV-1 and MERS. And we're collaborating with Matt Freeman at the University of Maryland to test that. There's a number of other connections here with proteins involved in RNA processing that are linked to translation as well. So we think these translation inhibitors could also have effects against SARS-CoV-1 and MERS, and hopefully with SARS-CoV-3 in the future. And we're looking into that right now. Here's a couple of connections I just wanted to highlight that we only see with MERS. These are blue edges here, and this is with NSP14. And we find a number of connections with proteins or factors involved in DNA repair or DNA damage response, including the MRX complex, MRA11 and RAD50, as well as one of the most well-characterized cancer protein of all time, P53. So the question is, what is NSP14 doing with these DNA repair, DNA damage, many of these cancer genes? And we're looking into this in more detail. So there's a lot of hypotheses that are being extracted from these maps from us and others around the world, looking for commonalities, but then also looking for viral specific connections as well. So as I said, in parallel, we're also expressing each one of these proteins and looking via microscopy, were they localized in the cell? And this was done in collaboration with Andrew Pedden in Sheffield and Robert Gross at the University of Freiburg in Germany. So we're transfecting these proteins in, localizing them. And for about half of the SARS-CoV-2 proteins, as I said, we actually had antibodies against the endogenous protein. So we could look in infected cells. And this was done in collaboration with Massimo Pallamrini at the University of Glasgow in Scotland. So we generated a lot of data, localization data through all of this. All of this data is available for anybody to look at. We looked at it and tried to summarize where we thought each one of the proteins were localized to. This is the data from transient transfection from SARS-CoV-2, SARS-CoV-1, and MERS, and where we think each protein is localizing to and which subcellular category. And the more blue it is, obviously, the more where you think it's localizing there. And then, as I said, we were looking also in infected cells with about antibodies for about half the SARS-CoV-2 proteins, and that's summarized here with these different colored boxes in red and orange and yellow represented here. So there's some overlap here with the transient transfection, but there's also some differences as well. But overall, there's a wealth of information that the community is now digging into to get a better understanding of the underlying biology behind COVID-19 infection. I want to highlight one connection here right now. It's a connection with one protein, ORF9b, that exists with SARS-CoV-1 and SARS-CoV-2, but not MERS. We find it's the only SARS-1 or SARS-2 protein localizing to the mitochondria. That's through transient transfection. And we also see it in infected cells. This is what this red box means, using the endogenous antibody. And this is the image of a localization of ORF9b in infected cells. And what you're seeing here is essentially a quintessential localization to the mitochondria. So I want you to remember this connection between ORF9b and the mitochondria, because I'm going to come back to it in just a couple of slides. So what we were also doing in parallel to the proteomic work and the localization work was trying to then go back to our original 332 genes or proteins to try to genetically validate them to see when you perturb them, do they have any effect on SARS-CoV-2 infection? And we took two strategies here. We again worked with the Pasteur Institute, Marco and Veronica, just fantastic collaborators over the last 7 months. And they used RNAi to knock down each one of these genes in A549 cells expressing ACE2. And again, using qRT-PCR as the readout for infection. And then in parallel, we collaborated with the company Synthego, in particular Jared and Kevin, where they did CRISPR knockouts in a different cell type, in CACO2 cells. And then these cells were sent to Chris Bassler at Georgia State University in Atlanta. And he did a very different assay for us, where he was infecting cells, then taking the viral particles off those cells, and then reinfecting other cells, and using, again, microscopy as a readout, using an antibody against the N protein. So we had two different assays, two different genetic perturbation strategies, and two different continents that were looking at our 332 proteins. And here's a summary of the genetic data. So in red here are genes that when knocked down by RNAi have an effect on infection. In yellow, this is the CACO2 data. In orange is where you see it in both cell types, including you see sigma R1 here. So we see some similarities, we see some differences, but overall, we have 73 genes that we've genetically confirmed out of the original 332 interacting human proteins, which I would argue is a pretty significant chunk of the proteome. And as you all are well aware of, there's many other genetic screens that have been going on, genome-wide CRISPR screens, two that have been come out in a Cell recently. There's another one that we've done in collaboration with Andreas Pushnik, which will hopefully come out very soon as well. And then Charlie Rice has had a couple of papers on bioRxiv, including this one where they also just targeted our 332 proteins, and they got a significant list of genes that was a good overlap with what we found as well. So there's a lot of genetic data now coming online that we and others can start to put together in very creative ways to get at more of the complexes and the pathways that are being impinged upon in the course of infection. But here's a summary of the genes that we derived through our genetic studies by focusing on the proteomic hits, the 73 genes. So each blue node is one of the 73 genes. And then I'm showing you here connections to these diamonds. These are physical connections to coronaviral proteins, either SARS-CoV-2 here in red, SARS-CoV-1 in orange, or MERS in yellow. And I want to highlight one particular connection for you right now. It's a connection between ORF9b. Remember I showed you it localized to the mitochondria. Well, ORF9b, both from SARS-CoV-1 and SARS-CoV-2, we find physically associated with TOM70, and TOM70 is a mitochondrial chaperone. So this is consistent with the localization data that I showed you previously. And I remember I told you at the beginning of my talk, we had a subgroup that was focused specifically on the structural characterization of SARS-CoV-2 human protein-protein interaction. So it's the Structural Biology Consortium within QCRG. This is being led by two great young scientists, Oren Rosenberg and the QBI fellow, Clem Verba. They put together a group of about 60 researchers, mostly trainees, mostly students and postdocs, and they're working together in a really unparalleled, exciting way they've set up these biochemical and structural biology pipelines so that they can characterize these connections that we're identifying through our proteomic work and genetically validating through our genetic analyses. And this is from 18 different labs here at QBI. And this group was able to successfully get a cryo-EM structure of TOM70. It's a 3 angstrom structure of TOM70 binding to ORF9b. TOM70 is in green. ORF9b here is in orange. And you can see that ORF9b is actually tucking itself up into the active site of TOM70. It's being like a cage up there. It's being caged by TOM70. And the question is, what is ORF9b doing to TOM70? Well, TOM70 is a chaperone. It chaperones proteins in and out of the mitochondria. So maybe ORF9b is using TOM70 to get itself into the mitochondria, and or maybe it's manipulating the chaperone of other human proteins in a way that's beneficial for the virus. And we're looking into both of these theories in more detail. A deeper look here at the structure. It's mostly hydrophobic with 36 out of the 38 ORF9b residues interacting with TOM70. And very interestingly, when we look at our phosphorylation data, we find that 2 residues on ORF9b at S50 and S53 are highly phosphorylated during the course of infection. And those are 2 sites tucked up here in this cage. And if these sites were phosphorylated, there's no way ORF9b could be interacting with TOM70. So we think that there's a couple of phosphorylation events that's inhibiting this interaction, which is then fighting off infection. We think we know what the kinase is, and this group is very close to getting a cryo-EM structure of the kinase actually binding to ORF9b. And just to go a little bit more detail into this ORF9b, we call it the shapeshifter. It's a very fascinating protein because when it's by itself, like it is here, it forms a dimer. So these are 2 copies here, what I'm showing you of ORF9b, and you can see it's primarily comprised of these beta sheets. But when it's by itself, it has a very different configuration. You see, it's primarily this alpha helices when it binds to TOM70. That's incredibly rare for a protein to have such drastically different shapes depending on its cellular context, depending on which proteins it's interacting with. And maybe this is why these viral proteins, these coronavirus proteins, are so deadly, is maybe they have so many different shapes, and that's how they can impinge on even more complexes and pathways during the course of infection. And I also really love this example because it combines the proteomics, the genetics, the cell biology, the biochemistry, the structural biology in a way to identify what we think is a key node for SARS-CoV-1 and SARS-CoV-2, and we think for SARS-CoV-3 as well. This structure has now been handed off to people like Brian Scheukat here at UCSF, and he's making predictions about drugs and compounds that could come in and bind and hopefully inhibit the detrimental function of this hijacked interaction. There's been a number of other QCRG papers that have come out over the last couple months. This was a paper that came out last week, led by Ashish Manglik and Peter Walter. They work closely with the QCRG Structural Biology Consortium to derive these nanobodies and structurally characterize them to inhibit the interaction between ACE2 and the spike protein. And a similar analysis was led by Jim Wells as well. I should also point out there's a number of campaigns in QCRG identifying compounds that would bind directly to the viral proteins, and we're characterizing those in a number of different ways as well, in the context of viruses and biochemically and biophysically and structurally as well. And I just want to end here by talking about some silver linings here that I see that have come out of this particular pandemic. Obviously, it's been a huge tragedy, but one of the great things that we've been able to see is that people have really worked together to expedite research on COVID-19 and SARS-CoV-2. And I would argue there's serious problems with the scientific enterprise that they've always bothered me. And a number of problems here are related to the fact that the individual gets rewarded more often than a group. And to me, that often impedes scientific progress because you're selecting against collaboration. That's particularly pertinent for young scientists. They come in and they don't want to collaborate because the system is set up where one person gets an award, one person gets a grant, one person gets tenure. And what we've seen here is when we throw all of that, those silos out the door in the context of this pandemic, we see how fast we can move. We see how fast we can move when we can collaborate collaborate across different laboratories, across different institutions, and between the academic world and pharmaceutical companies. And the question I have is, why aren't we working like this all the time? Why don't we keep this in place so that we're better prepared for the next pandemic? But why aren't we also working on all diseases in this fashion? Why aren't we working on breast cancer and Alzheimer's and Parkinson's? And for me, as a scientific community, one of the challenges is let's keep this infrastructure in place. Let's keep this spirit in place as COVID-19 goes away so that we're not only prepared for the next pandemic, but we're working on all diseases like this, because it's not just more productive, it's more fun, I would argue, as well. All right, so I acknowledge a lot of collaborators that we've had around the world over the last several months on our SARS-CoV-2 work. Here's a picture of my lab that was about a year ago, which seems like 10 years ago. Obviously, this was before the shelter in place. Just to highlight a few people, I had alluded to Dave Gordon, a postdoc in the group, who's now an adjunct assistant professor, and Gwendolyn Zhang, a technician. They spearheaded our protein-protein interaction work back in January on SARS-CoV-2. Mehdi, a postdoc, led the phosphorylation work, which I briefly alluded to. And then I also want to highlight Danielle Swaney and Ruth Huttenhain and Robin Cake. These are three fantastic mass spectrometrists that are affiliated with my group, and they're instrumental not just for this work, but for all the work that we're involved in. So it's a great pleasure to work with them and all the collaborators around the world. And finally, I'd like to thank you all for your attention. I'd be happy to take questions.
Thank you very much for the excellent and very insightful talk. So we have time for questions, and I'll be going through them one by one. One first, perhaps general question to start with is, anonymous attendee was wondering, how does collaboration between academia and pharma work with respect to the ownership of drugs that actually end up working? Are there any guidelines, agreements with respect to managing conflict of interest, ownership issues, things like that?
Well, normally, of course, there are. You know, we've worked with different companies of various sizes over the years. What was really great here in this pandemic, those IP issues for many of our relationships were just thrown out the window. We just said, well, look, we need to find solutions here. Let's just work together, and we'll worry about the IP issues later. So that was incredibly refreshing. Obviously, I don't think that can stay in place as we tackle other diseases, but I think people are starting to look at what's happened over the last several months when academia and pharmaceutical companies truly work together when a lot of the restrictions are thrown out the window. So the challenge is to kind of revisit how things are set up and then how can we change things, as I said, going forward. And we've learned a lot of how we can work together more efficiently. So I hope a lot of those lessons get implemented into these relationships in the future.
Thank you. Next, also somewhat general question is about, can you elaborate a bit more what are computational methods that are used most in these papers or in your group?
Well, for each data set, there's obviously computational methods, right? So we have different, we have different search algorithms that we use for the mass spec, and then we have different algorithms that we use to derive protein-protein interactions from the mass spec data. For obviously the structural biology work, there's algorithms that are used, there's computational tools that are used to derive these structures. Same thing for the genetic analysis, same thing, different tools for the microscopy work that I talked about as well. And so those are individual analyses, obviously, of each data type, but then where it becomes more challenging but more interesting is developing tools that allow you to integrate the different data sets together from the different technologies. And that's where we're at, you know, and many groups are at, you know, the bleeding edge at, and not just putting together two types of data, but maybe more types of data in intuitive ways. So we learned a lot here over the last few months in terms of how to do that. And we're very excited now to be applying this to other disease areas or other biological problems. In particular, how to put these different data types together in intuitive ways.
Thank you. Then another question is about that proteins can undergo different PTMs. How consistent are these across different cell types?
Yeah, it's a good question. So we initially published our work from Vero 6 cells. Since then, we've generated phosphorylation data sets in three human cells. And the data is quite consistent. Obviously, there are differences, which are interesting, just as interesting as the commonalities. And there's other groups doing similar things as well. There was a paper in Molecular Cell too. And there are a lot of similarities, but there are a lot of differences. And you know, we got to look at all this data in the context of you know, maps, pathways, complexes, and then figuring out how to overlay other types of information, like protein data and genetic data, or pharmacological inhibition information, to help prioritize what to look at. So the more the better, and the challenge is to put it together in intuitive ways so that we can generate hypotheses about the biology of the virus, which hopefully point us in valuable therapeutic directions.
So kind of a follow-up, how much do you think COVID pathogen endogenous protein interactions identified in HEC-293 cells will be consistent with those that would be identified in lung cells?
So we generated maps now with A549 and NHBEs and THP1s. The data is quite consistent, and which is satisfying. And the genetic data I showed you, right? We generated the PPI data in HEC-293 cells, but then we genetically validated it in lung cells, A549 cells, and CACO2 cells, two different cell types. And this allowed us to, you know, functionally validate 73 of these, which to me gives me great confidence. The data we're deriving in HEC-293 cells, even though it's not the most relevant cell type, although the virus seems to be infecting every organ we have now, but on the surface, it may not be the most relevant cell type, but we're validating these using orthogonal approaches in more relevant systems, not just genetically, but through structural approaches like cryo-EM. If you get a structure of an interaction, I guarantee you it's going to be valid. So we're getting a lot of structures of interactions that we derive from HEC-293 cells.
Super, thank you. Then another question is about, while you are researching the SARS-CoV-2 medical treatment, does your team try to map out the mutation of the future, you know, version 3 type of virus, and how to deal with it?
Yeah, that's a good question. We've actually been interacting with Harmeet Malik at the Fred Hutchinson at in Seattle. He's, you know, world's expert on zoonosis, and he's been looking at some viruses that have been sequenced in bats, and he's got some predictions about, maybe this virus will be the next one, or this one. So he's given us a couple viruses, and we're starting to gear up now to generate maps of viruses that he predicts. Along those same lines, we now have bat cells from Adolfo's group here in the lab, and we're generating protein-protein interactions in bat cells. So hopefully that could also be informative in terms of the zoonosis process, and being more predictive about what's going to be the next problematic virus as well.
Okay, thank you so much for answering the questions. Really appreciate your keynote today, and that you were able to join us and present the latest research in identifying the cure for COVID. So thank you very much again.
Thank you very much for the invitation.